everyone, my name is Kesten Bozinovic and I'm an EMAP student at Georgetown University. Today I'm going to be talking briefly about a portion of my summer internship at NOAA where I primarily worked with NOAA's Office of Oceanic and Atmospheric Research with people including Dr. Jessica Cross, Dr. Dwight Gledhill, and Dr. Gabriella Kitch. As part of my internship, I participated in a 3.5-week-long research mission to monitor ocean acidification in Southeast Alaska. In collaboration with NOAA's Ocean Acidification Program, the NOAA Pacific Marine Environmental Laboratory contributes to the observations of ocean acidification in Alaska coastal seas effort. As part of this, the lab maintains ocean acidification hydrographic survey along the continental shelf of the Gulf of Alaska. Although originally planned as a monitoring effort done every 4 years, the survey has been delayed since 2018 due to COVID, and it finally took place August of this year. The intended goal of the mission is to track acidification conditions along the regional southeast Gulf of Alaska system in order to support cross-agency efforts in maintaining regional time series, updating regional biogeochemical algorithms, and ultimately leading towards the development of an ocean acidification indicator. You can see in Figure 1 with the sampling stations map that the 2015 dataset is focused on offshore lines. This was the first monitoring mission, and the original plan for the 2022 mission was to replicate those sampling efforts. However, unexpected bad weather forced us to primarily sample the inside straits instead. While we can't make direct comparisons to the original monitoring effort, the data is still valuable, especially since coastal measurements have a more direct relation to local communities. Overall, 150 casts were deployed throughout the mission, with real-time sensor data along the water column to produce physical oceanography profiles that included temperature, oxygen concentration, salinity, pH, beam transmission, and fluorescence. We also performed hydrographic sampling at 70 of those casts, collecting data such as dissolved inorganic carbon, O2, nutrients, and eDNA, which will help both calibrate the sensor results and expand our understanding of the regional carbon system and its vulnerability. Many of the preliminary results we found based on sensor data are specific to the individual straits we sampled at. However, we can make a couple generalizations. First, we were able to sample at 4 offshore lines during weather windows throughout the mission, and compared to the 2015 data, the water was overall both colder and fresher. This may be due to a number of factors, one of which is that there was a marine heat wave in 2015, and it's also been found that the Gulf of Alaska is trending towards freshening over time in the long term. The data collected from the inside straits indicate that the entire interior of southeast Alaska is flushed with corrosive bottom waters. However, this is preliminary data relying solely on pH sensor data, which can be variable, so we have to wait to compare those results to the bottle data to determine the actual corrosiveness of the water and the saturation horizon. Wrangel Island is also of particular interest because it was found to have both pH and oxygen levels lower than expected, and it was the only site with an observed instance of shallow water hypoxia. Since this mission is part of an ongoing monitoring effort, the first future direction of this project would be to continue sampling in the area, capturing both the offshore sites from 2015 and the interior sites from this mission. I would also recommend working with the local communities in Alaska to inform the location and types of sampling for future efforts within the inside straits. Although the data is all preliminary at this point, until the bottle samples can be analyzed, future efforts should aim to determine the relative contributions of the biological pump, anthropogenic CO2, and other natural sources of variability in the carbonate system within this region. Another significant portion of my internship throughout this summer that I didn't address in this presentation was focused on a policy evaluation of carbon dioxide removal, or CDR, technologies. A potential future direction that works as a bridge between this research and the larger scope of my internship would be to further investigate Glacier Bay National Park as a potential natural laboratory for ocean alkalinity enhancement, since the source waters for Glacier Bay, fed from Chatham Strait across Icy Strait, were shown to be corrosive, while according to the pH sensors, there wasn't any corrosive bottom water present in Glacier Bay as would be expected, and it's been shown that the glacier system releases a lot of alkalinity. Finally, I want to acknowledge Dr. Gledhill and Kitch from the Ocean Acidification Program for being great mentors for me with the policy evaluation work I did this summer. And I also want to give a huge thank you to Dr. Cross, the rest of the PMEL staff, and to the entire crew of the research vessel Rachel Carson. Without all of their enormous efforts, the Gulf of Alaska Ocean Acidification mission would not have been possible.